just going to go ahead and do a quick introduction, and then also share with everyone that we are, along with doing the fundraising for the Whitebird Clinic, we are also have started a GoFundMe for our chapter of Synapse at the U of O. And I will go ahead and put that into the chat box so that people, if you would like to donate or would like to check out the GoFundMe or share it with others, you'll be able to do all of that. And so the next individual that we have speaking is Matt Smear, PhD. And he is an assistant professor in the Department of Psychology and the Institute of Neuroscience at the University of Oregon. Dr. Smear studies the neural mechanisms of olfactory function in mice. Mice have an excellent sense of smell. Much of their genome encodes odorant receptors, which consist of over 1,000 genes, and a large portion of the brain processes olfactory information. These neural features support a rich repertoire of olfactory behaviors, and the Smear lab interrogates olfactory function with a battery of psychophysical tests while manipulating and recording neural activity with genetics, electrophysiology, and imaging. With no further ado, Dr. Smear, the floor is yours.
All right, thank you. So, Daniel encouraged me to give something of a overview of the sensory system I study, which is the sense of smell, which obviously originates in the nose, as painted here by Magritte. And so when we think about smell, usually we incorporate smell into our lives in two ways. Does the person next to me smell good or bad? And how does this lovely beverage or food strike me in terms of my flavors? And this is a typical way to think about olfaction for a measly human, but most self-respecting mammals also use their sense of smell as a sense to give them spatial information. This is a picture taken from National Geographic in which the photographer dragged a pheasant across a field and had a dog track it while wearing an LED collar, and then took a long exposure photograph to follow the dog's path. And you can see the dog has this very structured back and forth movement across the trail, which it uses to track this pheasant target. And so that's the aspect of olfaction that I study most of all. But I felt for this audience, I would give more of the introductory stuff about olfaction, just to give you, the context, and maybe just talk about my stuff a little bit at the end. By the way, please feel free to ask questions as I'm going through. Just unmute or go ahead and throw it in the chat. I'll see. I have the chat window open. I love to be interrupted. Don't feel rude. And this is a lecture I usually give to students in my intro class. So if there are things that I said, words that I use, or anything that is unfamiliar to you, please feel free to just ask. Okay. So if we're thinking about the sense of smell, first thing to take into account is that odors propagate in a turbulent way. So they travel through the air, basically like clouds of smoke, just ones that you can't see. And so this has major implications for the way our olfactory system is structured. In some ways, olfaction is mainly an aerodynamic problem. So the air containing the odorant molecules have to flow through this network of tubes in your nose, shown here in this imaginary section through a person's skull. And you can see these structures extending off that are called turbinates. And the yellow region indicates the olfactory epithelium. That's where you have receptors that can actually pick up odorants. So we have this complex bendy structure in our nose It's even more elaborate in species that are use their olfaction more, such as dogs. This is a 3D model of the interior structure of a dog's nose. And what you can see here is this very complex foldy structure, kind of spongy structure. And the part colored yellow here is full of olfactory sensory neurons that can respond to odor stimuli. You know what? I opened the wrong PowerPoint here. So let me get the right one. I'm sorry. Apologize for this. That's the one I want. There we go. Okay. Loaded up my graduate lecture instead of my undergrad lecture. That one's even more bizarre. Now, please let me. Okay. What the heck? Do I have too many things open? Oh my goodness. I'm so sorry, you guys. Why is this? What is going on here?
While you're doing that, Randy, you probably want to mute yourself.
Oh. I'm going to. I've never seen, you find new ways for your computer to mess up every time you use it. And I have really never seen PowerPoint fail in that particular way before. So it's a treat for all of us.
I experienced the same thing today for the first time. So yeah.
What's going on?
Um,
There we go. Okay, thank you. And so these aerodynamic structures probably meant to absorb as much molecules from the air as possible. So starting from the aerodynamic aspect, where we have objects in the world that are emitting odorant molecules, those are received in the nose and stimulate a particular type of sensory neuron called olfactory sensory neurons that extend these little hairs into the mucus surrounding the epithelia in your nose. And so these are the only neurons in your body that are directly exposed to the outside world. So what are they exposed to? The molecules that are present in the ambient air we breathe typically are emitted by objects, and objects are typically chemically complex. So most of the things we smell, like a rose or a cup of coffee, what you're smelling is not just one molecule. All these sources emit hundreds of different volatile molecules that can fly through the air. And yet we don't perceive coffee as 275 different things. Our brain takes all those molecules, receives them, and synthesizes them into a usable percept. Another important fact about odorant stimuli is that it's still very hard to predict what a molecule will smell like based on its molecular structure, and it's often surprising. So these top four, so you don't need to know chemistry to appreciate that these molecules are very different, these structures. And yet, despite their differences, they all smell musky to humans. Now look down here at 5 and 6, these two molecules differ by only one little line segment right here. And yet one of them smells like urine and the other has no odor for humans. So just relating structure of molecules to perception is still a problem that our field is really actively engaged in. Jump ahead here. Oops. So the receptors that are used in these olfactory sensory neurons are encoded by what are called odorant receptor genes. And these were discovered in the early 90s by Linda Buck and Richard Axel at Columbia University. And so the discovery ultimately won them the Nobel Prize. And what they discovered were these odorant receptor genes. And they did so in a really clever way. I'll jump through this fast. One is that, as I told you, the odorant world is very complex. So a functioning olfactory system has to be able to sense lots of different molecules. And so because there are so many odorous molecules, that suggests that there should be a lot of genes for odorant receptors. So we can have lots of different receptors in our nose that can bind to lots of different molecules. Secondly, they assumed that the odorant receptor gene should be expressed in the olfactory epithelium. And then lastly, they had made an assumption about what kind of gene it would be. I won't go into this, but basically at the point where they did this experiment, it had just been learned the gene sequence of a typical type of receptor that's used throughout the brain and body called a G protein coupled receptor. And they assumed that the odorant receptors would be that kind of receptor. And so they did some genetic tricks and were able to find a large family of genes that were G protein coupled receptors that were expressed in the olfactory sensory neurons in the nose. And so their work allowed them to discover 18 different genes that they assigned as odorant receptors. They showed that these receptors were only expressed in the olfactory epithelium. And they estimated that there would probably be 100 of these genes in the genome of a rat. I should have said that was the species in which they did these studies. But they were off. So as Biandra said in my introduction, the largest gene family in every mammalian genome, including that of humans, is the largest gene family encodes odorant receptor genes. So this little bar graph here gives you an idea. So the bar indicates how many functional odorant receptor genes different species have in their genome. And so humans, although we think of ourselves as kind of having an impoverished sense of smell, we even have like 400 of these odorant receptor genes. And then other primates, rodents have way more, like 1,000. The one I work on, the mouse, has over 1,100. Cows about the same, horses. But the best one is the elephant. The elephant has 2,000 odorant receptor genes. Okay, that's 5 times more than us. So what does that even feel like as a perception? I wish I had a way of knowing that. Another interesting fact about the odorant receptor gene family is that it's extremely diverse across individual humans as well as populations of humans. So what this study is quantifying is the similarity of the olfactory receptor genome among different ethnic groups. And you can see there's some clustering of here. But the point I want to emphasize is that in a study where they looked at, so different humans have different combinations of functional odorant receptor genes and non-functional. So it's really true. The world smells different to every individual human. A genomic study showed that in a sample of 5,000 people, not one of them had the exact same set of functional odorant receptor genes. So the diversity among different individual humans is really pretty staggering.
Can I ask, is there a way, I mean, there's no way to know, but is there research on both in humans and across different animals of how to understand that different levels of the smells perception inside of us? Or is that just something subjective that is, forever?
Yeah, so a lot of people are working hard on this. And, as with like most things in science, machine learning has proved to be really useful, like training neural network algorithms on a large set of molecular structures and a large set of perceptual, like people's words they use to describe the odors. But I mean, we can't even reconstruct it to a basic level, let alone to be able to say like, okay, this person doesn't have that receptor. They're going to, that smell is going to feel a different way to them. There are a couple isolated examples. So there's one receptor gene, which when mutated in a human causes sweat to smell less bad. So basically, if you have this receptor, sweat smells really bad. And if you don't, it smells less bad. There's also a genetic difference that explains why different people have different taste in cilantro. So there's an odorant receptor that's responsible for the intensity of a person's perception of cilantro.
Can I ask a question?
Please do.
I'm wondering what you might have to say about the relationship between a diffused brain injury,
Yeah.
cellular level, and partial loss of smell, or just a change in your smelling abilities.
Yes.
And
So
I'm assuming that means that's either the genes or the receptors, I forget what you called them, as a result of the injury may have been damaged, which would certainly account for an impediment in smell, which leads me to wonder, to what extent can that be rehabilitated, if at all?
Yeah, great question. So the olfactory sensory neurons are some of the most regenerative populations in the brain, because like they are really exposed to the outside world. So they die a lot, even under the most healthy of circumstances. And so when one has an injury that kills them, they tend to grow back in a few months.
Okay.
But why someone, cases where someone doesn't lose smell but just has a change in their smell perception, those are extremely interesting cases, and I really couldn't give you a good explanation for why the perception of smell changes. One possibility is that some of the receptors in the nose die and others don't, and so then the signals coming into the brain are different. But I suspect that often it relates to injury in olfactory areas of the brain, which is what I'm going to talk about next.
Okay. Thanks.
So,
that was good.
So the olfactory system was a source of fascination for the earliest neuroscientists. This is a drawing by the great Ramon y Cajal, who's like the godfather of modern neuroscience, in showing some of the structures in the olfactory system. So what I want to emphasize right now, these represent those olfactory sensory neurons I was telling you about. They send wires called axons into the surface of a region called the olfactory bulb, where they make synapses in these like little ball structures that are called glomeruli. So basically, this is where the nose wires into the brain. The olfactory bulb differs in size across different species. So in humans, the olfactory bulb is here in the bottom frontal part of the brain, the ventroanterior. In comparison to a mouse, this is a mouse's brain from the same angle, looking from below. You can see a mouse's brain has an olfactory bulb that's a lot larger relatively. Interestingly, though, if you look across mammalian species, we basically all have the same number of neurons in our olfactory bulb. So even though next to the rest of our brain, our olfactory bulb looks small, it's actually about the same size as that of a mouse. And so going back to the glomeruli, I was telling you about where these systems wire in. A really kind of my favorite example of how the brain wires up is the way the nose connects to the olfactory bulb. So what happens is you have all these cells that express different receptors, and they're just scattered throughout the whole nose. They're all over the place. But somehow, from all that mess, that chaos, the brain has to say, those receptors are the ones that are active. That's the molecule that's out there. And so the way the brain solves that problem is extremely elegant. So this image comes from another paper by Richard Axel's lab. So we're looking in a mouse's brain, and the mouse has been engineered so that all the sensory neurons expressing one receptor turn blue. So all these dots here are olfactory sensory neurons in the nose. And you can see these black lines coming off them. Those are their wires going into the brain. And what you can see is that they all converge in this one little spot on the surface of the bulb. So you go from this very wide distributed pattern in space to somehow knowing that you need to send your wire to this one little spot that we call a glomerulus. So, sorry. And then on from the olfactory bulb, they send their axons into a large network of regions that basically cover the whole bottom of a mouse's brain. So I would say probably like maybe even a quarter of the whole mouse brain seems to be olfactory structures. So humans, on the other hand, have relatively smaller olfactory cortical areas. So compare this, which is the whole bottom of the mouse's brain, to this little tube here running along the ventral surface of the human brain. And so you might wonder, how are we going to understand how a sense works when the part of the brain that is important for it is so deep buried in the brain? Well, as you know, one of the techniques of modern neuroscience that we can use with humans is called functional MRI. And that takes advantage of the fact that oxygenated blood has different magnetic properties than deoxygenated blood. So when the brain is being scanned, the metabolically active areas of the brain change their fluorescence or their intensity, which allows you to pick up patterns of brain activity. And so trying to do smells under these circumstances is interesting. This is a schematic of one of this type of experiment. So the person goes into this fMRI scanner wearing a mask with tubes carrying odor, which are in turn connected to a device that controls airflow really precisely. And with that in valves, you can say, okay, I'm going to blow air out of the purple vial right now, and the person's going to smell that, and I'm going to watch what happens in their brain. So go through this a little bit quickly, but one thing that's been shown is that it doesn't take an odor to activate these cortical regions. Even just sniffing of clean air activates the olfactory cortex. So this kind of underscores the fact that what I said earlier, olfaction is kind of an aerodynamic problem, how air flows. And so the brain represents not only what molecules are in that air, but also the flow of the air itself. And that's something I've worked on a lot over the course of my career.
Question, so what does that mean? Does that mean it's a different signal in the brain, sometimes detecting odor, sometimes detecting airflow? Or does that mean we don't have enough resolution to know what's the difference? Because I would think an odor and an airflow would have to be different to an extent.
So the way I think of it is like the air is the medium, the air is like the piece of paper that you're writing on. So you can't really extricate the writing from the paper. The writing needs to be on a medium somehow. And so I think of the airflow as kind of like the way the brain represents where the piece of paper is, and whereas the odor driven signals are more like the words. So I guess what I'm saying is I actually don't think they're at a perceptual level, they're different. Your brain kind of has to know how the airflow is going through your nose in order to interpret what the molecules are doing.
I guess that makes sense. I never thought about that, but
so yeah. So that's like my main interest in the big picture now is in general, how does a brain integrate information about how you're moving with how you're sensing? When we look at the world, our eyes don't just freeze like a camera. Our eyes are always moving around a little bit. And those tiny movements that you can't even resolve, in normal conditions are necessary for functioning vision. And so that reflects the fact that our brain represents not only the light that's coming in our eye, but also how the eye is moving in the orbits and in the head.
That's a
So
good
I think of
example.
airflow as kind of the analogy to that.
Cool. Thanks.
So let me jump through the fMRI part. So I talked about how we have a little bit fewer odorant receptors than other species. So, and I think kind of the received wisdom among humans is that we're kind of bad at smelling compared to other animals. But a series of studies actually shows that it's not that simple. So there was a paper where the researchers synthesized other papers where the smell had been studied in 17 other mammalian species, including several bats, monkeys, a harbor seal. Must be fun doing odor experiments with harbor seals. But the take-home message from this is that if you test all these different species in their ability to detect odor, small amounts of odor. So you do a test where you say, okay, here's a little bit of odor. Can you smell it? Yes. Okay. Here's a little bit less. Here's a little bit less. And progressively reducing until you find the level where the animal can no longer perceive the stimulus. And so each of these dots here represents one species. So down here is the one outlier. These are dogs. But then up here are all these other mammals, including pigs and rats. Their dots are, if anything, above the brown dots that represent humans, meaning that actually most mammalian species are less sensitive to odors than humans. So we have this very sensitive olfactory system, but you might wonder, okay, but is that actually important to our lives? I would say look at the very mixed history of the spice trade, the effect that had on human development and culture. Most of the Christopher Columbus and those kind of folks' discoveries were not motivated by exploring the world. They were motivated by let's get the spice more quickly. And so, why am I talking about spice? That's a taste thing. Well, you may be familiar with the fact that our perception of taste is really an illusion. So what we're actually experiencing when we think we taste something is largely mediated by air flowing through the back of our mouth and in through the back of our nose, and those molecules traveling rather than from the external world, traveling from inside our mouths. And so that kind of olfaction is called retronasal olfaction. And I think that highlights one of the ways in which anosmias, whether inborn or caused by some brain injury, I think this is a big reason why people with anosmia report higher levels of depression than people with, let's say, blindness or deafness. Even though we think of olfaction as playing a subsidiary role in our life, it has this plays this role in this illusion that's so important to our quality of life.
Randy, look.
Yeah. Can you repeat what you just said again one more time? I'm sorry.
From how long?
Just the last few thoughts that you had expressed about the
illusion segment, which I think all of it's an illusion. Oh.
Actually, my first thought about all this was, does this explain why dogs eat their own crap?
Yeah. No. So dogs have another olfactory organ that humans don't have anymore. Most mammals have this other organ that's called the vomeronasal organ. And it is like a little pump in the roof of the mouth that basically pulls, and the function of it is so that you can sense molecules that are not volatile, that aren't airborne, bigger molecules that don't fly. You need to actually contact in order to sense. So this vomeronasal organ, I think, is actually the reason why your dog likes to lick things and eat its own crap and things like that. Is largely because of this organ.
Interesting.
I talked to a woman who does dog training, working dog training. She's like the person where they call to testify when there's some kind of dog related evidence in a criminal case. And she was telling me dogs do this thing she calls vomeronasal licking. So in her experience, she has the pleasure of looking for cadavers in boat, like going out in a boat to look for submerged cadavers. And she says, anytime a dog licks the water, she knows there's a body. And again, the dog is licking because whatever is being emitted by that cadaver, a lot of it is not going in the air. A lot of it is, stuck in the water. And so the dog, by licking that water, actually brings the water up to its vomeronasal organ that it can use to sense what's in there.
Is the central organ for smell, I forgot what you called it, is that part of the, I take it that's near your brainstem?
The olfactory bulb?
Yeah.
No. So the olfactory bulb is actually out in the front of the brain by like the cortices that are important for decision making and things like that. So it's actually at the very anterior tip of the brain. So it's kind of, and that kind of makes sense, right? Because
like up here.
It's like kind of just above your eyes.
Okay.
Why would it be at the front of the brain? Probably because that's the first processing structure that evolved in the brains of, the ancient ancestors of vertebrates. So, it's so ancient. So one of the ways olfaction is unique is that most senses have to go through a structure called the thalamus in order to get to the cortex. Olfactory information goes straight from the olfactory bulb into the olfactory cortex. Let's see. So Wendy's asking, isn't there a relationship with anosmia and neurobehavioral deficits? The thing I'm aware of that's most associated with anosmia is depression.
What is anosmia?
Anosmia is like smell blindness.
Oh.
Yeah. Let's see. I guess I'm running over. I could talk all day. I love to talk more, answer more questions, but I guess I've elapsed my time. Can I show one more thing before I go?
Yeah, absolutely.
Okay. So I said I would show you a little bit about the work from my lab. As I said in the beginning, in addition to telling us what odor is out there, for many animals, the sense of smell is important for telling you where something is. And so that's the aspect that I'm really interested in. I want to know, how does the mouse follow its nose to find the cheese or the peanut butter or whatever? Okay. So I'm interested in how the aerodynamics of how the mouse moves its nose around and sniffs for odor. So this is, oh my God, this is the a video of an experiment from our lab. So here's a mouse. We have a computer that is tracking points on its body. So it's tracking the tip of the nose, back of the head, and the center of the mouse's body. What's happening here is odor is coming out about here and about here. same odor, but at different concentrations. So the mouse's job is to go toward where there's more odor. I'm really obsessed with sniffing and how, this aerodynamic aspect. So that's represented in this movie in two ways. So when the mouse is inhaling, these dots and lines are blue, and when he's exhaling, it's pink. Also, there's a sound you're going to hear, hopefully, that it's a higher tone when he's inhaling and a lower tone when he's exhaling. And this is slowed down by 4 times. They do this really fast. I could watch this all day. Sorry, I better. I guess I kind of do watch this all day. Guess I'm blessed. Anyway, so that what we're trying to understand here is, how does the mouse's brain not only sense the odor, but how does the mouse control its movements in order to sample that odor in the best possible way? And so the big picture that we'd like to connect to is, again, the fact that our senses are very active. We're always moving our eyes. In order to touch something, we have to, palpate it with our hands. The sensory systems are not like passive cameras. They're very active. And so that's what I'm interested in.
Thank you. Thank you for
Very
sharing.
cool.
And then just really quickly, there was a question in the chat from Patty that said,
Yeah.
is asking, can things be done to regain smell blindness?
That's a great question. So, when smell blindness is caused by damage in the nose, that I think usually recovers because those neurons are constantly growing back. But if there's something else happening deeper in the brain, I don't know of any way to rehabilitate smell. I bet there are ways, but it's just not something that I think people have explored yet. But it's a great question. I wonder, like for example, my mom can smell a few things, but largely has lost her sense of smell. So I wonder, if she trained herself to learn what different molecules feel like, I wonder if she could restore that function.
Yeah, absolutely. It sounds like an amazing research question that someone will be able to explore in the future. I want to say thank you so much for presenting with us today. It was really fascinating. I find the olfactory system and the gustation system fascinating. If you don't mind, a couple of things. We've been asking speakers if they are comfortable with sharing their slides with us,
Sure.
so that way we're able to distribute it to the members in attendance. If that's something that's okay with
Absolutely.
you. Perfect.
Daniel, I'll send it to you. Will that work?
Sure. Thank you, Matt. That was great.
Perfect.
Yeah, no problem.
All right. Thank you so much. I really appreciate it. And well, hopefully you'll be able to stick around for the other panelists today.
I can stick around till 2, so I'll hang till then.
All right.
And if any of you guys have other questions about what I've talked about, my email is just smear@UOregon, so not too hard to remember. So yeah, I welcome you to, if you have any weird questions about smell or anything like that, I love that stuff. So please feel free to email me.
Perfect. Thank you so much. And I will put that email address in the chat. Just really quickly before we move on to our next speaker, I know that time has been kind of wonky, but the next speaker, if you have to go a little bit into 2 o'clock, that's totally fine because there's an entire hour block that's dedicated to one speaker. And so we can definitely move and shave off things there. And then just a really quick plug. If anyone in attendance has a GoFundMe account for another really awesome fundraising or fundraising team or event or program, go ahead and put that link in the chat. And what we're gonna do is when we send out an email to everyone that's been here, we're gonna be including those links and contact information